Get started. So we have an open slot this afternoon. So Boris Schreiman will be lecturing from 2 to 3:30. Public lecture starts only at 5:30. So we thought it would be a good idea since there was a huge amount of data and a lot of different kinds of models that were discussed in Ted's talk, that we could have a tutorial on sort of stochastic models of gene expression. Is this something people would be interested in? So we'll actually go hands-on and we'll see how these models are done. Okay, fine, great. So in that case, we'll break for tea at 3:30, come back in here at 4 o'clock, and we'll just run a tutorial from 4 to 5. And then the public lecture is at 5:30. Yeah? Okay, just so you all remember. Good. Okay, so like I said, based on Ted's original topic, I thought one of the things that would be useful to discuss and present in great detail were some broad introduction to the different types of cells in the world, and of course this talk that I'm going to give is not just introduction. So similar to yesterday, what I'm going to do is to spend about half the time giving a broad introduction to things, and then spending the second half giving a specific example of a question that one could work on a topic like this. Okay, so what I'm going to talk about is the evolution of complex cells, and by complex cells, it's a little biased, so that should say eukaryotic cells. Right? But if people don't know what eukaryotic cells are, then this is sort of an approximation. I should say morphologically complex cells. This beautiful picture, by the way, is done by an Aboriginal artist named Shorty Changarai, and this has some meaning if you know how to read these things. There's a whole story going on in here, but when I first saw this, it really sort of captured for me two different things. So one was I just looked in here and I saw all kinds of things that you might see inside a cell, right? So I mean, you all see that, right? So but the other thing I saw was something else about timescales. So I thought it brought both those things together. So here we go. So these are the timescales I'm going to talk about. So 14 point something billion years ago, you have the Big Bang, and very soon after that, the first stars, they coalesce into galaxies, and so on. The solar system is not that old. It's 5 billion years old. The Earth is 5 billion-ish years old. The surprising thing is that the first cells on Earth that we know about from fossil evidence are 3.5 billion years old. So I mean, I don't know if you think that's a short time or a long time, but the fact is that very soon after the Earth formed, you have fossil evidence of cells on Earth, and this should be quite surprising. It has implications for origin of life on other planets. Yeah. What is the error bar for that? So this, so if you speak to this comes with so I would say this is it's likely to be more recent than this, but people pushed it back to this much. Okay. So anyway. But certainly this one has very, very little error bar. So 2.5 billion years ago, something happens in the geological strata, which is very easy to date, and you can date it all over the world in different kinds of rocks and so on. That's when oxygen comes into the air and sea, and you get oxygenated minerals. Okay, that's called the Great Oxygenation Event. Anybody know why that happened? Cyanobacteria. So this is the first example of sort of life-driven climate change on the planet. You know, we're doing similar things today, but bacteria did it a long time ago. They invented photosynthesis, and therefore they released oxygen, and that actually sets the stage for the kinds of organisms we are. We depend on oxygen to derive energy to run our cells. Okay? So I'm going to talk about the time frame from here onwards, which is when you go from having just bacterial cells in the fossil record to having more complicated cells, which are the eukaryotic cells. This beast called the last eukaryotic common ancestor is just a fictitious organism. It's just an abstraction, but that dates to about 1.5 billion years ago. Here's some other timescale people are interested in. Around 700 million years ago, you get the first animals in the fossil record. So that's the history of life on Earth. This is the history of our understanding of cells. So as I said yesterday, one of the big ideas of biology is that all life is made of cells, and this might be a very important thing. Now, it turns out that, so of course, I don't know if you've all seen these pictures, but you should. So Van Leeuwenhoek looking through his microscope, Robert Hooke looking through his microscope at the same time, but completely independently, find out, in Van Leeuwenhoek's case, he finds out that there's all kinds of single cell, well, he didn't know, but little animalcules running around in water. Okay? All kinds of myriad forms. Robert Hooke looking at a tissue actually of plants, and he sees that the tissue of cork is made up of these little segmented regions which look to him like prison cells. So he names them cells. Right? So cells are named after prison cells. And so very, very quickly everybody realizes that all living things on Earth are made of cells. It wasn't clear before that big organisms like us had some sort of common origin with free living microbes. But the fact is that's what it is, and that's the origin of the cell theory. I'm going to skip all that history, but if you come to 1900, which is again not a long time ago, if you asked a practicing biologist what are the big distinctions in biology, they'll say that's easy. There's unicellular organisms and there's multicellular organisms. Right? And the sort of tacit assumption being that all multicellular organisms share a common ancestor and all single-celled organisms are somehow more ancient than that. It had to wait till the 1940s for the advent of the electron microscope before people realized that this was wrong. Okay? So it turns out that there is a fundamental distinction in all of life, but it's not unicellular versus multicellular. It's something called prokaryote versus eukaryote. Prokaryon means nucleus. So prokaryote are cells without nuclei. That's the shorthand. Eukaryotes are cells with nuclei. You've all studied this in the textbooks. I'm going to tell you that it's not as simple as that. But anyway, the electron micrographs ended up showing that there were two types of cells, one which had all kinds of complex internal structures and tended to be a little bit bigger, which were the eukaryotes, and the other ones which were small, tended to be morphologically rather simple, the prokaryotes. Okay, so this particular distinction was so stark, there were no intermediate forms between prokaryotes and eukaryotes, that people started asking, did they come from a common ancestor, which was the ancestral form, and is there some way you can come up with a scenario for how these cells arose from those cells? Now, thinking about this actually dates back quite early, but the person who really pushed a certain model of eukaryogenesis, the origin of eukaryotes, was Lynn Margulis. Famous today, but at the time she had a very difficult time promoting this particular idea, which seemed crazy, which was that a eukaryotic cell actually arose when two different prokaryotic cells, which used to live freely, ended up in a symbiotic relationship. Okay? And it wasn't a symbiotic relationship where they were in the same environment. It was endosymbiosis, which means that one of those creatures actually went into the other creature and set up residence over there. Okay? So it does seem crazy because, remember, yesterday I talked about Darwin's model of life as a tree. In this case, you don't have a tree-like behavior. You have two branches coming together to make a third branch. Right? So it's interesting. It must be a rare event, but it does happen. And these organisms, we now have very, very strong evidence that the mitochondria, which are the respiratory centers of our cells and all eukaryotic cells, used to be free-living bacteria. In fact, their closest relatives outside today, the alpha proteobacteria, are very much like mitochondria. And there's lots of lines of evidence for this. And I can discuss them with you later. But one of them, the strongest one being the mitochondria, come with their own DNA. And the other one, which really clinches it for me, is that the outer membrane of mitochondria is at a molecular level identical to the outer membrane of alpha proteobacteria. There are proteins that are found only in the outer membranes of these and only in the outer membranes of those bacteria. So that really clinches it. Okay. In the 1970s, people started to sequence DNA. So this was fine. So people thought, okay, this is good. And what we need to explain is more about this particular divergence. Carl Woese was one of the first people to use the idea of sequencing DNA as a way to make a really large classification of life. So he ended up sequencing the DNA corresponding to ribosomal RNA, which is something that's common to all living things, as far as we know. And he did the first sort of big data experiment in biology. He just crunched the data. And he ended up finding something pretty shocking, which was that there aren't just these two different things, which it would appear to be from the microscope's images, but actually there were three different kinds of living things on Earth. The DNA of these three things are very, very different from each other. And at the moment, we still don't really understand how all these three pieces of life fit together. I'll describe a little more about it later. But it turns out that the prokaryotes, the small, simple cells, themselves break up into two completely distinct lineages, which are called the bacteria and the archaea. And the eukaryotes are somehow a third lineage, and we don't know how these three relate to each other. Okay? So that's where we are. So now we come to the present decade, And you can ask the why now question. Why revisit this particular question of where these complex cells came from? And the answer is sort of obvious. So for the first time ever, we have data on thousands of genomes. We're not just focusing on model organisms, E. coli, yeast, mouse, and so on and so forth. But we have genomes from natural samples from the ocean and so on and so forth that really cover a lot of this diversity of biology. So we have enough data to talk about these distinctions. The second thing is, and this is very important, we actually have a good understanding mechanistically of how cells work, what makes them work from the inside. Right? So these two things having reached a certain stage, maybe it's a good time to revisit the question of how these complex cells arose. Are there any questions about this sort of history of cell biology?
Are there any archaea that have taken any traces of archaea in eukaryotes?
Yeah, eukaryotic DNA is a sort of a mixture. No. So it's a mixture of both. So if you take any gene in a eukaryote, its closest match is either in bacteria or in archaea. So we contain a lot of DNA signature from both those things, which is rather confusing. So it turns out that a lot of the enzymatic machinery comes from bacteria, and a lot of the DNA reading and the ribosomes and so on, all that informational machinery comes from the archaea. Turns out that our membranes, though, look more like bacteria than archaeal membranes. So it's sort of very, very bizarre mix of both those things. Yeah? Yeah?
What about plastids?
So plastids were also bacteria, cyanobacteria, that took up residence in eukaryotes. Now that's a little less mysterious because it's happened a couple of times. So we've sort of seen that happen.
So in these, let's say between bacteria and archaea, is it that some mechanisms are different, or is it sequences are for different classes?
So bacteria and archaea are so different from each other that some people propose an independent abiotic origin of these two things. Right? In other words, their common ancestor was not really a living cell. That's how different they are. They have lipids. So bacteria, I forget which one is which, but bacteria have ester-linked lipids, and archaea have ether-linked lipids. So it's hard to imagine how these two go together. The ribosomes are very different. All the machinery is just you can't really mix them up. Right? It's not just a minor sequence variation. It's a rather fundamental difference. The genetic code is the same, by the way. Yeah, that was yeah. Sorry. Viruses are very messy. They come from everywhere. You can't really follow them. They change very fast. So I would not put viruses on a tree-like history. They just, I mean, you saw earlier, lambda can come in and go. They can pick up samples. They can take DNA from you and combine it with bacteria. So yeah, I would not go there. There. I'll describe it. Yeah, I'll describe that. Back there.
Yeah, you said that the three kingdoms, bacteria, archaea, and they have a different kind of DNA. I mean,
I didn't say that.
No, you said not DNA are different in those.
The sequences are slightly different. Yeah.
So my question is it possible to read out quantitatively those sequences to have the distinguishedness of those?
Yeah, so that's in fact exactly how they did this. They just clustered the DNA sequences and found 3 distinct clusters in sequence space. Last question. The genetic code is the same for mitochondria and for us.
How does this
Play into the story?
yeah.
Yeah, well, so it turns out that mitochondria, so it's just, again, a point I brought up earlier. It turns out that all living things use the same genetic code, except for 1 or 2 species. So this dates back to a sort of pre-cellular origin of the genetic code, right? It could be. Genetic code arose, and it's not clear that it arose inside a fully functioning cell, right? But anyway, so mitochondria, I'm not saying that the DNA is different. The DNA molecule is the same, okay? And it's not even that the genetic code is different. It's just that if you look at the entire protein sequence, there are flavors of proteins that are found in bacteria, there are flavors of proteins in archaea, and there are flavors in eukaryotes, and they tend to form different clusters. Okay? So I agree this is very messy, but that's because that's how it is. Okay? So let me just give you the data, the way that looks. So this is what so can you imagine trying to do fossil cell biology, right? So what you're doing is you're cutting some sort of deep stratum, and you're trying to find micron scale structures in the electron microscope of these rocks, right? So it's a very difficult thing. Very few people do it. There are big error bars over here. Nevertheless, so this is an example of what counts as one of the earliest microfossils. You see sort of membranaceous structures here. The data is not much more than that. They can't even find the correct elements in these rocks to say that there were living things in there. But the rocks definitely date to several billions of years old. It gets a bit better when we come more recently. So starting around 1.2, GA is giga years ago or billions of years ago. So starting around 1.8 billion years ago, if you look at various rock strata, primarily in Australia and in China, you start to see these kinds of things in the rocks. The scale bar is, I think, 10 microns, so several tens of microns. And these things clearly look like membranes, and it's sort of hard to make a non-biological origin of many of these structures. What this graph shows you is interesting. You can't quite see it because of the resolution, but every one of these different textures corresponds to a different cell type. And what you see is that in the early days, there are cell types that don't exist today, but as we come closer to the present day, most of the cell types you find in the fossil record are part of the same types of cells that are alive today, which sort of makes sense, right? The only thing I want to say about this is that there is a time scale which is actually very well established. We have eukaryotic fossils going back at least 1.8 billion years. Okay, so good. Now let's talk about the present day. What are the types of cells that are alive and around today? Yeah.
There wasn't some Cambrian explosion like
In eukaryogenesis?
cell business where you sort of see a lot of diversity and then it went down or something.
So yeah, so even if such a thing were there, it would be very difficult to make a claim from two rock bores from Australia and China. Okay, so that's the only problem. So the Cambrian explosion happens with microscopic organisms. These are animals and you find it across the world. So we don't have statistics on this yet. This is as good as it gets. This is pretty good. This is pretty good. Okay, but there was, in some sense, a huge diversification of eukaryotes. If you look at all the eukaryotes today, this is a phylogenetic tree. It's made by Parfait and colleagues. This is the best tree that we have so far. What they did was, unlike Carl Woese, who took just one particular type of DNA, they took many different protein sequences. They concatenated them, and they ran phylogenetic reconstruction, and they calibrated these using what's called a molecular clock hypothesis, also against fossil evidence. So these gray bars are places where we can date certain fossils, and we can say that this particular lineage existed at that time, and so you use that to calibrate your molecular clock. So this is what this says, and this sort of also agrees. There's sort of several billion years. There's 1.5, 2 billion years. So not disagreeing with the direct fossil evidence. So this phylogenetic tree, there's two important things. One is that all the eukaryotes that are alive today, by definition, will have a common ancestor. Any set of organisms growing in a tree-like process, if you just take everybody alive on a certain day, they will be a common ancestor. That's the common ancestor there. That's the last eukaryotic common ancestor, right? The most recent common ancestor. It doesn't mean that there weren't eukaryotes around before, right? But those didn't lead to any lineages that are still alive today. Important point. That orange dot is meant to represent this mitochondrial endosymbiosis, this mysterious process we don't we don't know exactly when it happened, but it certainly happened only after oxygen came around. That green dot is the chloroplast endosymbiosis leading to plastids. So everything on this side are the photosynthetic eukaryotes, the plants, the algae. Okay, but the important thing to realize is there's a huge diversity of eukaryotes. This group over here, the opistheconts, forget all the names, they consist of us, the animals, and basically the fungi. So yeast and humans are both part of this lineage. In fact, this lineage is rather recent. And the reason that yeast is such a good model organism in biology is because it's almost identical to humans on this kind of scale. Right? When somebody says something is conserved from yeast to humans, that's not quite surprising. If something was conserved from plants to humans, that would be more surprising. Okay, that really dates the ancestry. Yeah. How would we know? But anyway, there's been many different kinds of endosymbiosis subsequent to this, but all the subsequent endosymbioses have been eukaryotes of other eukaryotes. It's very strange. Okay. But the original bacterial one, but there are bacterial parasites today that there are bacterial parasites of other bacteria. They live inside other bacteria. The parasites, I mean, the boundary between parasitism and endosymbiosis is not completely clear. Amoeba, Dictyostelium is over here. There's some ambiguity about whether it's on the same side of the tree as us. Actually, most people think that they are on the other side of the tree, and therefore the common ancestor of amoeba and humans would also be that old. The excavates, these strange cells like Giardia. You have plants, the photosynthetic eukaryotes, ciliates like Paramecium, and in here you have the diatoms. I mean, there's a huge diversity. Okay, so you might think, okay, so eukaryotes are very, very diverse, and where do we go from here? But the fact is, if you actually look at the cell biology inside the cell of every eukaryote alive today, they all more or less look like this. Right? This is very surprising. So this is why I was saying, so to say that eukaryote means you have a nucleus and prokaryote means you don't have a nucleus, that's just the tip of the iceberg. What it really means is if you are a eukaryote, not only do you have a nucleus, you also have this thing around the nucleus called the endoplasmic reticulum. You have a Golgi apparatus. Of course, you have mitochondria. You have floppy membranes because you make sterols like cholesterol. Okay? You have other kinds of what are called organelles, which are little compartments that sit inside the cell and are specialized to do different things. You have these extracellular structures called flagella, and either you have them today or you lost them, but all eukaryotic lineages had them ancestrally. So the point is that all eukaryotes are this complex. No prokaryotes are this complex. Right? So it becomes difficult to try and make the connection between them. Okay? So one way to try and do that is to go down to the molecular detail. So you say, fine, this is what it looks like under the microscope, but what are all the molecules that are responsible for making all these things? And let's see if the molecular evidence can somehow allow us to reconstruct history. So this is how we do that. So again, this is a sort of schematic of the inside of a eukaryotic cell. This is the outer membrane. It's called the plasma membrane. This is the nuclear membrane, which is contiguous with the endoplasmic reticulum. This is where all the protein synthesis happens. Proteins get processed in this organelle called the Golgi apparatus, and then it gets sent all over the cell. Okay? So the way things move around in the cell are through these small vesicular carriers, these membrane-bound carriers that can package proteins inside or on the membrane itself. And the way a vesicle is made is through a process of budding and fusion, which I'll describe in greater detail later, but it involves a series of proteins which are called snares, rhabs, coats, tethers, and so on. Okay, I'll describe these in slightly more detail later. But the point is, all the organelles inside the cell are set up by making membranes and by moving stuff between membranes on vesicles. Okay? And these are the proteins that do all this. So what we could ask is, look at all the eukaryotes that are alive today and just see, do they have all these proteins? If all eukaryotes have these proteins, then, or most of them, then the assumption is that the last eukaryotic common ancestor also had those proteins. Okay? Or even more specifically, if one side of the tree and the other side of the tree has the same protein, even though they've been lost in certain lineages, you can infer that it was present in the last eukaryotic common ancestor. So by doing this kind of reconstruction, Mark Field and Joel Dax have shown quite conclusively that the last eukaryotic common ancestor had all the proteins that you need for eukaryogenesis. In other words, LICA was already a full-blown complex eukaryote. It was not by any means a simple organism. Okay? So all the interesting stuff in eukaryogenesis happened prior to LICA, which again makes it difficult for us to reconstruct what was going on. Okay? So how did LICA in the first place become a complex beast with a bacterial ancestor? Yeah? So that's the question. Everybody happy with that? So let me make the question even more clear. There was some ancestral cell. Yeah?
So, two questions. One is, are they similar looking homologous proteins in prokaryotes?
Yeah, good question.
B, I guess this idea that what you see today, if it has everything, then it must be present in the common ancestor.
Yeah, so
It's a little biased, right?
No, well,
It says that it is conditional on survivability.
oh, yeah. No, but no. Fine.
You outcompete the guys which sort of were.
Yeah, no question. But they're all dead. We both agree on that. Yeah, fine. So lineages, unfortunately, the lineages that gave us evidence of simpler eukaryotes are not around right now. They may be in the deep sea. We don't know where they are. Okay. The point about prokaryotes, it's very true that prokaryotes, for example, have actin homologs. They have tubulin homologs. They have dynamin homologs. Right. They even have coat protein homologs. I'm not claiming that bacteria don't have these. I'm only claiming that LICA had all these. That's one important point. Also, prokaryotes are a little more difficult to deal with because horizontal gene transfer is rampant among prokaryotes. It's very, very rare among eukaryotes. So if a prokaryote has something, it's not clinching evidence of ancestry. It might have picked it up more recently. So don't rely on that.
So viruses don't shuttle around
well, not the sort of these core things. It's very rare. It's very rare. I mean, they do, but it's rare. That's
Yeah. So are you focusing on things which are required for trafficking and compartmentalization?
yes.
Is that the most fundamental difference between eukaryotes and
This is my bias. I think what makes a eukaryotic cell is the fact that it has internal compartments, and therefore I'm focusing on trafficking. You could also, of course, focus on metabolism, right? But that ends up taking you to the electron chain on mitochondria, and that ends up being of prokaryotic origins, right? So that becomes a little more difficult. But of course, polymerase you could also look at. There are many other things that you could look at. I'm focusing on this because I'm talking about compartmentalization. Other people looking at eukaryogenesis could look at other things. For example, the origin of sexual reproduction, or the origin of a certain type of DNA organization in the chromosome. Right? Those would also be perfectly reasonable types of molecules to go after. And people do. I'm not showing that here. It's a good question. Yeah? The size of LICA. See, we don't know anything about, so LICA LICA is like this mitochondrial Eve who lived in Africa. There's one woman who is in all our ancestries, right? It's not that there was only one woman around at the time, right? But she's the one who contributed DNA to everybody alive today. It's just a fact of population genetics, right? So this is an abstraction. There was a whole population of things alive at that time, right? And we'll hear more about that in this afternoon's lecture. Okay? So to compress the question again, so how do you go from a morphologically simple cell to a morphologically complex cell? How do you add a new type of compartment inside a cell? Right? That's the one thing I'm focusing on over here. And you could think of two ways to do this. One way is Lynn Margulis's explanation, where she says, well, one way to get an entire compartment is just to have a whole cell and to take the whole cell into your membrane, and then that thing, like the mitochondrion, becomes an independent organ of you, sort of enslave it. Okay? Now it turns out that, of course, mitochondria and chloroplast came like that, but all the other ones didn't come like that. Okay? All the other ones arose de novo inside the cell. So you need some other explanation of this. Okay? So maybe a better way to think about this, how do you make a new cellular compartment? Rather than Lynn Margulis's quote, I like this quote by Carl Sagan. They happen to be married at one time. So Carl Sagan says, to make an apple pie from scratch, you must first invent the universe. And this thing is filled with meaning. But one way to think about that is, to understand something today, you have to look at where it came from. Actually, what he's talking about here is the hubris of physicists. But anyway. So let's see where things came from. So here's the best model we have of eukaryogenesis so far. It looks the following way. The last universal common ancestor of all living things dates to around 3.5 billion years ago. This is the present day. Ancestral cells were very likely prokaryotic, and so this sort of cloud are the bacteria and the archaea. We don't really know how they're related. Bacteria don't really grow in a tree-like manner, so I have this bush over here of bacteria and archaea. Somehow, somewhere, close to the global oxygenation event, one of these cells is going to become the ancestor of all eukaryotes. It was not a special cell at the time, and it had no idea about its coming greatness, right? But that is the ancestor. There is a process of eukaryogenesis where all kinds of complicated things happen. But as we heard, many of these lineages could have easily died out, right? They're not present today. Therefore, the last eukaryotic common ancestor is much more recent than that, right? This last eukaryotic common ancestor is 1.5 to 1.8 billion years old based on the phylogenetic trees I showed you. This is 2.5 billion years old. So you had 1 billion years to make a eukaryote. Okay? So that's what we want to explain. So what happened on this branch? Right? If we'd had examples of intermediate forms, that would have been great. But we don't. Maybe we haven't looked hard enough. Right? Maybe they live in the deep sea. Right? Or maybe they all became extinct. But we can look at the molecular data again. And this is actually quite important. So again, present day, 3 billion years old. Let's see what actually happened. Well, this, I've already told you this story, but I'll go over it again. You had a prokaryotic ancestor. You had the oxygenation event. Subsequent to that, there's the mitochondrial endosymbiosis. Okay? Subsequent to that is the emergence of compartments. And I'll explain why this happens after that in just a second. You get more and more compartments, increasing complexity, until you get this last eukaryotic common ancestor, which is as complicated as any cell living today. If you look in the genomes of eukaryotes, you see the following pattern. It's very striking. First of all, there are some protein varieties that occur only in eukaryotes and not in prokaryotes, although this is not very important for the argument. What is very important is Prokaryotes tend to have 0 copies or 1 copies of many of these types of proteins, whereas eukaryotes tend to have 10, 20, 30, 40 copies. Okay, there's what's called a gene family expansion. And this gene family expansion we can date because it's in the lineage of our own genomes. These are gene duplications. This is a phylogenetic tree, a sketch of that. And these are gene duplications that happen in my own genome ancestrally. It's not a tree of species. So I can actually trace this gene family expansion by looking at the genomes of plants or of animals or of any cell that's alive today. So this we have very, very good data on. So here's something that's a nice pattern. This gene family expansion happens at the same time, basically on that long branch, as the emergence of compartments. Okay? It's just a correlation, but maybe these two things are related. So here's our current best explanation. Bill Martin makes this point very strongly. You can read this paper in Nature. Very, very nice calculation of how much energy the mitochondrion actually brought to the eukaryotic cell. It's not based on membrane area, by the way. It's an interesting and subtle argument. But his point is the following. You have a prokaryotic ancestor. The mitochondria gets enslaved. This cell now has a very large energy budget to splurge. It has a surplus. Whereas prokaryotes are limited in the size of their DNA, this organism is no longer limited. And therefore, its DNA could just expand. Okay? Not in any directed fashion. You just have a lot more genes. Subsequently, many of these genes get repurposed or purposed for new function. Okay? That gene family expansion coincides in time with the diversification of compartments that we get from the fossil record. So I've now just set up the following question. How do changes in the genotype, in terms of gene family expansion, lead to the emergence of new compartments? That's one way to frame a question. Okay? So, yeah. This one? Yeah, so fine, very good. So I would direct you to this paper, but it goes the following way. So it's expensive to maintain DNA. It's expensive because you have to keep DNA and replicate it, but it's also expensive because if all your DNA codes for proteins, those proteins take money to make. Okay? So you can actually work out how much this costs. So the nice thing is when you have, so let's assume that mitochondria are the same size in their DNA as any prokaryote alive today. They have a few thousand genes, okay, ancestrally. When a mitochondrion enters the other prokaryotic cell, okay, it brings its DNA with it. Imagine that the prokaryotic cell just gets stuffed with mitochondria. That just looks like a collection of prokaryotes. It's not particularly interesting. Then something funny happens. The mitochondria start to lose all their genes. And huge numbers of genes go away from the mitochondrion into the nucleus of the host. We don't know if there was a nucleus, but it went into the DNA of the host organism. Okay? That means that this cell now has a huge number of extra genes in the host genome, and just the energy producing genes present, by the way, I didn't mention this, but mitochondria are present in tens to hundreds of copies inside a eukaryotic cell. So you have a lot of energy producing genes in large copy number, and you have the rest of the genome in single copy. So per gene, you have a lot of energy. That's how that works. Okay? It's a subtle argument, but read the paper. It's very, very convincing. Does that explain that? Yeah. Sorry, I'll just take that one. Oh, the mitochondria duplicate on their own. They have their own processing and duplication. Yeah, good question. Yeah.
So is there evidence that all the extra genes from mitochondria actually went to the DNA of the host?
Yes, very, very clear. Because you have many different lineages of eukaryotes which have lost different amounts of mitochondrial DNA. You can actually see mitochondrial DNA going into the host. If you put a gene in here, at some rate, you'll find it in the nucleus. I mean, it's all very clear. Okay, so this is the big setup. This is sort of the background, right? So now I'm going into the sort of specific case study just like I did yesterday. But I thought, because yesterday I talked about genetic networks, I would just do a quick recap with some lessons from there. So everybody happy with prokaryotes and eukaryotes and the diversities? Yeah? It's entirely possible that the mitochondria came in as a parasite. So that, I mean, there are other parasites today which behave in a very similar way.
So I mean, so did it actively sort of produce the compartments? Is it possible that it might have?
It's possible, but you need to frame that as a hypothesis that one can test. Yeah? That's the whole problem. Okay, so I'm just going to do a quick recap of some of the stuff that we talked about yesterday. Part of the lesson over here I'm just trying to show is that the kinds of models that one can make, the modeling frameworks actually apply across many different kinds of problems. Okay? So I'm going to do a recap of models that people have been using to understand gene networks, and then I'm going to repurpose those models to try and understand these sort of trafficking networks. Okay? So this is very, very quick recap. So Stuart Kaufman was one of the earliest people to make sophisticated models of gene networks, and his models had the following features. So he described every gene, just like I talked about yesterday, as a binary variable. The gene is either off or on. Right? You now understand the nature of this approximation. And also the genes can talk to each other through all kinds of logical functions. Right? And then the state of the gene expression can change over time based on these interactions. Okay? So based on, and so this is a discrete in time, discrete in state model, and in this particular case it's deterministic, so I'm just going to walk you through this. So imagine that you have some binary vector that describes the state of 4 different genes. It's actually nice to rewrite that as a sort of binary integer. Right? So I'm just going to, this whole thing is actually it's the binary number 12, but you add 1 to it because 0 becomes 0 otherwise. So there are 2, 4, 8, 16 states, and you're going to describe them as 16 integers. Okay? And this is the state number 13. So now because of all these interactions, some gene turns on, those genes turn off, fine. So you transition from that state to some other state. And it can happen again, and it can happen again. Okay? So this will keep going. So what's interesting actually is that in this case what you see is that starting from these 2 states you end up at this core, but once you're at the core you keep oscillating back and forth between them. Right? So these are the types of oscillations that we saw yesterday as well. The other interesting thing in this particular model is that if you start at other states, they end up going to a completely different core. Right? So of all the 16 states that this particular network is capable of, right, there are sort of 2 different basins. Both the basins end up going into some sort of oscillatory state. Here's a 2-state oscillator, here's a 6-state oscillator. Right? And of course it's possible that it would end up in a steady state. I just haven't shown that over here. So in general, these kinds of discrete in time, discrete in space models end up, you could use a binary model to explain the more complicated kind of thing that I talked about yesterday. Okay? But good question. Suppose we change the interaction. Suppose something happens in the environment that changes what's going on inside here. Right? Then of course these arrows will change. And then you could end up going to a completely different set of fixed points for the same system. Right? And this is the kind of thing people study. Okay? So let's talk a little bit about whether this is a decent approximation. And the sort of philosophy we will use is the philosophy that all models are wrong, but some models are useful. Right? So a binary model can actually be a very efficient description of the dynamics of a system. Moreover, it turns out, of course, it turns out to be much more easy to constrain a binary model. Right? Now, just because something is easy to do doesn't mean it's worth doing, but it turns out that it is worth doing. So here is a very nice paper by Chao Tang and colleagues where they made a binary model of the yeast cell cycle, both in terms of gene expression states and protein phosphorylation states. They just took all the known interactions between genes. Okay? And they had 11 genes, and they had all the interactions between them, and they just plugged into this binary model, and they ran it through. So every one of these things there are 2,048 points over here, right, 2 to the 11 points, and every point will update to a different point. And what you see over here, the point about the whole paper, is that there's a central trunk root, which turns out to be the actual root of the yeast cell cycle. And moreover, these other roots, these other possible initial conditions, most of them are actually quite close to the trunk root. So the argument that they make in this paper is somehow about the robustness of the trunk root of the yeast cell cycle. Okay? The point I'm making over here is that in this case, a binary model, which was experimentally determined, actually produced a prediction of the yeast cell cycle based on experiments that didn't know about that, and this came out of that model. So binary models can indeed be useful. Okay? But this is just the cell cycle. So it turns out that traffic is a bit more complicated than that. Traffic, by the way, is this process of vesicles moving around inside an organism. Here's a nice cartoon that came out in the Bangalore Mirror last year. So this is Bangalore, and this is traffic. You all know about it. Anyway, so why is traffic complicated? It's complicated purely because of the combinatorics. Okay? Even if you accept that a binary model is a reasonable approximation of the system, you don't have to accept that. But even if you accept that is the case, then it turns out that traffic systems are hugely complicated. The reason for that is the following thing. So here's the eukaryotic cell. Right? In this eukaryotic cell, you have all these compartments, and these compartments exchange matter by vesicles. Okay? And the composition of each of these compartments is different because they contain different proteins. And what proteins they contain makes the difference between being the endoplasmic reticulum or being the Golgi apparatus or whatever it is. Okay? So let's make a binary model. Every compartment I'm going to describe by a vector of zeros and ones, saying that maybe there are, in this case, 4 different types of proteins in a cell, right? And every compartment can have some subset of those 4 proteins, or it might have all of them. Right? So every compartment now becomes a binary vector. Okay? The problem is that in the previous case, the dynamics of the genetic network was completely autonomous. It depended only on itself. Right? But in this case, the dynamics of a given compartment depends on the presence or absence of all kinds of other compartments, which are sending it all kinds of matter. Okay? So the state space is actually rather large. Okay? And I'll get back to that point in just a second. Are there any questions about the setup of this? So all I've done is I've taken, this is purely pedagogical. I'm just trying to bring in one type of model and apply it to a different type of system. I'm just trying to bring in a Stewart-Kaufman type binary model, but apply it to a system which has many different compartments. And the update rules of the compartments will depend on its own composition, but also on the presence or absence of all kinds of other compartments in the same cell. Okay. So how do we make a model of this? You have to make an update rule. Right? Given the state of a system at this time point, what is the state of the system at the next time point? So here's an example of how you could make an update rule. There are many other ways to do it, but here's a nice abstraction. Right, remember, every compartment is now described by a binary vector. And that binary vector, in this case a 4-length vector, okay, we're going to put it on the rows and columns of a matrix. So these matrices, I'm going to describe in just a second, will give you the time update dynamics of the cell. Okay, that's what we're trying to do. So here's how we do it. This matrix is a so-called budding matrix. What it says is, if I am a compartment with a certain composition, what types of vesicles will I bud? And therefore, what type of material will go away from me? Okay? Along all the different columns of the matrix correspond to different compartmental compositions. There are 2 to the n compartmental compositions, 16 in this case. All the rows of the matrix correspond to different vesicle compositions, and a vesicle could contain any subset of the same molecules, right? So both vesicles and compartments are described by binary matrices. Inside the matrix, you have zeros and ones along any row or column. If you have a 1 in a particular row and column, it means that that particular compartment makes a vesicle of this particular composition. It's a nice way to do it. Now what? Well, that vesicle is now going to go into the rest of the cell, and it's going to bind somewhere, and it will deposit its contents in a different compartment. Right? So I have to make a rule for that as well. So again, I have vesicle compositions over here. I have destination compartment compositions over here, and I say this vesicle, if there's a 1 in that particular row and column, will fuse to that compartment. Right? So in this way, yeah.
Vesicles, how many molecules?
Can they pack? So vesicles can pack several tens of proteins. So 100 would be a good number. Yeah, but they may not. So how many flavors of proteins is the question? Yeah. So vesicles could easily pack 10, 20 flavors of proteins. It's not unheard of. Yeah. That's how stuff moves around inside a eukaryotic cell. So a eukaryotic cell, remember, is a. Oh, why don't proteins move freely? Okay, fine. So if you talk, we talked about diffusion earlier. So E. coli is a micron long. Okay, GFP will take somewhere between 1 second and 10 seconds to diffuse from one side of a cell to the other. Okay, eukaryotic cell starting at 10 microns could be 50 microns in size or bigger. If you were waiting for proteins to move under the force of diffusion, you would never get anywhere. They're just too big. So yeah, so exactly. If you want something to go across all, there are neurons that actually run from up here to down there, and you have to move things actively. That's one important point. The second thing is, a eukaryotic cell is not just a bunch of things mixing up, although I showed a picture of Bangalore traffic, so it might seem like that. But actually, there are different regions. Like there's one region of the cell which is the factory. There's the other region of the cell which is the packaging house, right? There's the third region of the cell where things are working. And so you want to move matter in a very programmed fashion. Now, when you want to do that, how does an individual protein know where to go? You'd have to have targeting apparatus on every protein. Rather than doing that, you make a particular targeting machinery, and then you just package proteins into that. So those are the two reasons. One is the size, the other is the spatial variation that you want to establish. Yeah? No, so lipids only occur in membranes. Okay, in a cell, you never find lipids except as part of membranes. Oh, vesicles. Okay, so it turns out that vesicles are pushed around. Okay, they tend to move on these actin networks, which are like railway tracks. Right, so they land on the railway track and they'll actually move in a directed fashion at a fairly high velocity. You can destabilize the underlying network. They will still get pushed around because eukaryotic cells have a lot of, so just the actin-myosin meshwork over there will cause a sort of high rate of cytoplasmic streaming, and so they'll reach different parts of the cell. But in a real functioning healthy eukaryotic cell, a vesicle will bind to a track and move to its target.
So what initiates this first? Some kind of signaling happens and then
The vesicle contains all the information it needs to bind. So it contains all the right targeting machinery. So once it's launched, it'll find the right partner, it'll get dragged to the right place and deposit its cargo. Right? So in fact, you can knock out proteins so that vesicles can't find their targets. Then they just accumulate and eventually the cell dies. Oh, then the vesicle will accumulate. You can account for it. This model is, by the way, I'm just giving an example of, this is an abstraction, okay? I'm not talking about using this as a particular very serious model of the cell, okay? Fine. Good questions.
So in the buds, you only have an entry. So you only have an entry for a bud which comes. Sorry. So one kind of compartment can only give one kind.
No, any number of vesicles. I mean, this thing could be filled with lots of ones.
But that's fine. But each one represents one compartment and one bud.
Yeah.
So a bud could come with two kinds of proteins.
So which proteins it has depends on the 2 to the n particular configurations over here. But one compartment can and will give many vesicles. The only constraint being that the vesicle, of course, cannot contain any material which was not on the compartment in the first place. Yeah? Okay.
A vesicle is also a matrix of that kind.
A vesicle is just a binary vector. Vesicle is just zeros and ones. The matrix is the interaction. The vesicle is just zeros and ones saying that it contains this protein or it doesn't contain this protein for the list of proteins you've chosen to look at.
Okay, so every compartment is just a vector.
Every compartment is a vector. Every vesicle is a vector. And these
GJK gives you a transformation matrix as to which
exactly.
compartmental states can transform into which.
Not transform, but produce which vesicle states. Exactly.
And the information, I mean, the actual construction of this matrix GJK.
Yeah. How does it happen? That is based on molecular interactions and molecular specificity, which if I have time, I'll describe at the end of the talk. But the fact is that proteins can bind specifically to other proteins. So a certain coat protein, I'll describe that later. I'll get to that. Yeah. Yes. Learning. No, only evolutionarily. These vesicles will go to their specific targets because they contain the correct sticky molecules on the surface. Not during the life of the cell. Because it contains the right, there's no such thing. It contains the right sticky molecules and the target contains the right complementary molecules, so the vesicle will bind to the target. It doesn't decide. It happens purely through purely physical interactions. Purely physical interactions. I'll get into that. I will get into that. Okay, so anyway, so it turns out this particular abstraction is actually sufficient to contain all these different molecular rules. So Sanjay asked, how do you actually populate this matrix with zeros and ones? That depends specifically on which coat proteins will make a vesicle, which adapters will pull the right molecules onto the vesicle. The vesicle will contain an address that tells it where the target is. Those are called snare proteins, which snare proteins there are. All these different molecular processes are actually how you fill in the ones and zeros over here. Okay? So, but the fact is that you can eventually come down to that abstraction. Okay. So now that you have a rule, we've done two things. We have described the state space of our model, which is the ones and zeros on every compartment, the set of all the compartments in the cell. We've described this sort of interaction step, which is compartments give vesicles, vesicles fuse to other compartments. This then leads to a time update rule in the following way. For example, suppose you have a compartment that contains some molecule, okay, that's labeled in pink because it contains the pink molecule. And there are vesicles coming in and there are vesicles going out from all the other compartments to all the other compartments. And some vesicle is taking the pink stuff out. Then over time, the pink stuff will get depleted and you end up with none of it. Right? Conversely, if the pink stuff is coming in and not going out, you'll turn pink. If something is coming in and not going out and you have it, you'll stay pink, and so on. So there is all these sort of simple update rules. Okay, so now that little bit about complexity. So remember what I said. I'm just putting all this out here, not to get into the great details of this, but just to tell you, even if you accept that a binary model is a very good approximation, okay, you can ask how complicated this model turns out to be, and it turns out to be extremely complicated. I just showed you an example of 4 molecular species, but a real cell will have hundreds of molecular species. So let's go with the number of molecules of the cell on this axis. Okay? On this axis, you have, I don't know why the axes are missing, I can see them on my screen, but anyway, on the vertical axis, you have the total number of states the cell can be in. Okay, that's the real state space of the system. Okay, this is logarithmic. Okay, this is ridiculous. With just 20 molecular species, you're already approaching, this is not even a number, right? What is 10 to the 83? It doesn't mean anything. Okay, this thing that I've drawn here tells you the number of states a given compartment can be in, and that's just 2 to the power n, right? So on a log linear plot, it's a straight line. Right, so the number of states a compartment can be in, which is 4 molecules, is 16, right? With, which one did I draw out over here? Anyway, so with 12 molecules, it's sort of 65,000. Is that right? No, with 20 molecules. Right? So the number of states a compartment can be in is quite easy, but a cell is a combination of compartments. So can you imagine, you have 65,000 possible compartments. How many types of cells can you make? Right? So really, even if you have a binary model, the complexity of trafficking systems is astronomical. It's ridiculous. Okay? Now this is where theory can actually make a difference. A theory or a certain sort of mathematical computation. So it turns out, and this is actually very, very important. So it turns out that if you want to search through the state space, remember for my gene network, I went through all the state spaces. In the Chow Tang example, there were 2 to the 11 states, and you made a little diagram of all those, right? In this case, you're searching through 10 to the 83 states, which is ridiculous. Everybody will agree, nobody's going to do that, right? But you can convert the behavior of this system, you can map it down to the behavior of a simpler system. Right? Essentially, you can reduce the size or the complexity of this problem from 2 to the 2 to the n, which is up here, to 2 to the n, which is down here. This is tractable. Right? And that comes through the application of a certain model reduction protocol. So this is the application of theory. You've made something simple. Of course, there's a trade-off. Right? This entire simplification depends on having the ability to do something called maximal clique finding on a graph. It doesn't matter what that is, right? But it's something that the computer scientists have been working on for a long time. So by applying some sort of powerful computer science machinery, you simplify a problem from ridiculous to tractable. Okay. Very quickly, I'm just going to show you what this kind of model does, and then go to a more concrete model where many of your questions will be addressed. So here's a specific example. This is the budding matrix. This is the fusion matrix. There is 4 molecules. Therefore, there are 2 to the 16 compartmental states and 2 to the end. So there are 2 to the 16 cell states. Okay, because a cell can contain any one of these compartments. Okay, and this is the particular budding and fusion matrix. You'll notice we populated it in some clever way. We're thinking about something, right? So Soumya is the one who did this. But here we go. Let's take this and let's start at all 65,000 states, because that's a small number, and see where it ends up. So this is what it looks like. Yes?
Do you want to specify how many compartments you have?
Yes, you do.
So what's that number that you think?
So in this graph, I've said 16 compartments maximum. Similar here. In fact, in this case, it can only be 16 compartments because there are 4 molecules. So, your question is slightly different. I'm saying that if 2 compartments have the same composition, their dynamics will be the same from that point on. So I just need to know whether a compartment of that composition is present, not how many varieties of it there are inside the cell.
Okay, so the state of a cell, I mean, there could be many compartments
Of the same composition, and I just identify all those with the same state. Yeah, good question.
simply asking whether a particular compartment type is present or not in the cell.
Exactly. Does it have a Golgi apparatus? Does it have an endoplasmic reticulum? Not how much membrane is involved in each one of these things. Okay. So for this particular thing, here's the result. There are actually not 65,000 things here. I've reduced things by applying certain symmetries, but that doesn't matter. So every one of these dots is a state of the cell, not the state of the compartments. Every one of these arrows, you can't see the arrowheads, but the arrows are the time updates. Starting from all possible states, you update, right? And then, for example, this is a state that just keeps updating to itself. It's a fixed point, but it's a very fragile fixed point. You have to start there to reach there. Here's a more interesting one. If you start at any one of these states, you end up in this pentagon, which is a cycle 5. It's a 5 cycle, right? So it's an oscillation. There are a few other 5 cycles here. Okay, this turns out to be a 4 cycle. Okay, take my word for it. But this one is the important one because the bulk of this initial conditions of the system end up over here, and that turns out to be a 3 cycle. So again, the point is that we've made a model, and that model now allows us to abstract out to this level. So when we started talking about traffic, it wasn't clear how to even picture these things, right? But now we have something we can work with, right? And now we can do all standard graph theory on this. Anyway, just so you know, that particular 3 cycle is actually this cell state, just for concreteness, right? It turns out to have exactly 5 compartments, and these 5 compartments have exactly these compositions using that integer labeling scheme, and it keeps updating between itself. So compartment type 4 becomes compartment type 14, that becomes compartment type 2, which becomes compartment type 4 again. These are the vesicle exchanges that happen. And an interesting point, at least I find it interesting, is in this steady state, which is the most robust steady state where most of the things converge, to keep the steady state going, you only need these particular interactions. Okay? The rest of the interactions are completely unimportant to keep the steady state going. So what are the rest of the interactions doing? The rest of the interactions actually make all these other conditions converge to the steady state. Right? So trafficking systems are robust, but that also means that a cell biologist who will only be studying the healthy cell which is here may or may not have the data to understand what's going on when the cell is perturbed. Okay, so that's all I wanted to say about that. But now let's get to the real model. Okay, so that was more pedagogical. So this might address some of your molecular questions. Okay? So before I get to this, can I answer any questions? Everybody's happy with it? So yeah. Well, no, that's going to take some time. Okay, so I'm happy to talk about that later. Okay, so any model of trafficking systems would have to account for 2 different types of things. So there are proteins that can diffuse in the cytoplasm. There's no question about it. Okay, not all proteins have to go inside vesicles. These cytoplasmic factors include these important proteins called Rab GTPases, but also many, many others, and that's shaded in pink over here. There are proteins, however, and these are sort of the shared goods of the cell. Every compartment has access to it. Right? But then there are also private goods. The private goods are what are contained inside a given compartment or stuck in the membrane of a compartment. Okay? These private goods can only be exchanged by vesicles because proteins cannot cross membranes. Okay? So the private goods are labeled in green, and I call them membrane-associated factors. They can either be these sort of transmembrane proteins, such as the snares, which I'll talk about in the next slide, or they can be cargo that's just stuck inside one of these compartments. Okay, and then there's all kinds of other general machinery that's required to bend membranes, cut membranes, move vesicles, the actin cytoskeletal tracts, and so on and so forth. So here's the actual molecular process by which a vesicle is made and is transferred. So let's start off over here. You have a compartment inside a big cell. There are lots of transmembrane proteins stuck on the compartment. There's also a lot of other proteins inside the compartment. Based on which machinery you have stuck over here, from the cytoplasm, remember the pink guys, you recruit what's called a coat protein. And the coat protein has 2 jobs. The first job is to actually physically bend the membrane, and this is a problem a lot of people are interested in. Okay? So it starts to make a bud. The other thing the coat protein does is it recruits a series of things called adapters, which pull certain of these cargo molecules into the vesicle, but not others. So in this case, that particular coat likes these 2 guys, but doesn't like these 2 guys. Right? So the vesicle that comes out actually is a subset of the stuff that's on the compartment. And that happens because of specific molecular interactions between coats and the cargo. Right? And in some cases, this is understood in molecular detail. In other cases, we don't know, but there's no reason to think that the explanation will be any different. Once you make a vesicle, it floats around inside the cell, or it gets on a track. When it reaches its correct destination compartment, there will be some sort of interaction between a label on the destination compartment and a label on the vesicle. This is why I was saying the vesicle contains all the information it needs to know where to go. If these things bind correctly, there's a fusion step. And what the fusion step does is it causes the vesicle to just deposit its cargo into the destination compartment. And that's it, you're done. Right? This turns out to be an energy-consuming process. You need to actually use ATP to reset the system. because these two things turn out to be stuck together. But anyway, this is the entire cycle of vesicle budding: recruitment, budding, and fusion. Question? Oh, yes, so one type of compartment can indeed recruit many different types of coats to make many different types of vesicles. Something like that. Yeah, although some combinatorial code might also be working. So, yeah. Yes, yes. There's only a few types of coat proteins, and those coat proteins will have specific interactions with certain adapters. So these are, you can actually find them, they fall into very clean families, and different species will have different subsets of these families. Yeah.
Related to the model and also the slide. So how do you remove, so in your transition matrix, right, you have 1, 0, 1, 0. Let's say. You want to go to 1, 0, 0, 0, so you are removing a certain kind. So it felt like if you're sort of budding and binding, it's all positive, right? You always sort of increase, so but here you sort of allow them to deplete away.
No, but even there you deplete. It could be that this factor is the guy that promotes the recruitment, right? But it could also be that this dark, or rather, let's say this purple factor promotes the recruitment of the coat. Then the purple factor gets sucked away. It's gone. So basically the purple factor is promoting its own release. So there are both positive and negative. Although it looks positive, there are both effective positive and negative interactions over here. It's a subtle point, but yeah. Okay? But this is the cycle, and you can read about this. So this is the bread and butter of any cell biology textbook. Okay? And how people discovered all this is deeply interesting, and I don't have time to get into it now. Okay, fine. So now let's talk about making a model with these things. It's a very different kind of model. In this case, I'm not going to make a binary model. I'm actually going to make a model where I put in protein concentration, specific interactions, and so on and so forth. Whereas the previous model was a pedagogical abstract model, this model actually puts in the biophysics of protein interactions. Right? So here's how we would make such a model. Let's say you have a cell, and it has a certain number of pre-existing compartments. These compartments are already in there. By the way, compartments can form out of vesicles de novo, and I'm not going to talk about that. Okay? There are all types of components in the cytoplasm. There are many different types of coats. There are many different types of RABs. Right? So those I'm going to index by some subset gamma. N sub C is the total number of coats, if you want to think about it like that. There are also a certain number of membrane-bound or compartment-trapped cargo, which are the blue guys, remember, and I'm going to index that with alphas and betas. Okay? In this case, the state space of the system, to specify it, you have to say, well, what is the size of the compartment? So this actually addresses Sanjay's question. In this case, there could be many compartments of the same composition. The size of the compartment is this W sub j. The size of a vesicle, let's assume it's fixed, okay? It's some W sub 0. On a given compartment of size W sub j, there are certain types of membrane cargo, okay? So this says that membrane cargo type alpha is found on compartment J at a number which is given by the variable x. Okay? So the state space of the system is basically all the different compositions of all the compartments. It's much more complicated than the last one. Right? So increasing the complexity of the state space means we have to somehow reduce the complexity of the update rules to make it a tractable problem. So I make a lot of assumptions starting from here. So here are the assumptions. I'll go through this in a little bit of detail. So here you have a compartment which contains these membrane cargo. Remember, x sub alpha j on compartment j. The size of the compartment is wj. Okay? Now some coat of type gamma comes to this compartment. And that coat could be recruited depending on the composition of the compartment. I'm going to ignore that possibility for now. Okay? That coat will now make a vesicle which contains some of the cargo that used to be on the compartment. Right? But which cargo does it like? Well, this becomes a bit complicated. So this thing says that coat gamma from compartment J now contains cargo type alpha, and that's the number that went there. Right? So of course, we're going to have to start to put some numbers in here. So let's say that the rate at which this thing happens, okay, will be proportional to some sort of prefactor, and of course the size of the source membrane. The larger the source membrane, the more the rate at which you can make vesicles. Right? But remember, vesicles are not sort of big flat sheets. They're curved, there's tension, things stretch, so it could be, just to make things slightly more interesting, let's assume it goes as some sort of exponent of the target area. Okay? Now, how do you actually recruit cargo? Right? So if cargo is present in amount x, so this is the concentration, the area concentration. X alpha j divided by wj is the concentration of the cargo in the compartment. What will be the concentration in the vesicle? So for that, I'm just going to make a little matrix, similar to the budding matrix I talked about earlier, but a little more concrete. This says that coat gamma likes cargo alpha with some sort of affinity, and that's how it packages it. Okay? So these two steps mean that I can package vesicles and I can move them out at some rate. Now, where do the vesicles go? Yeah. It's actually a stochastic process which will happen. It's a Poisson process in this model. In the cell, I don't want to comment about the real cell at this point. Okay, so now that you've made a vesicle of some type, vesicle of coat gamma coming from compartment J, right? It's actually a very good approximation. The vesicles don't float around in the cytoplasm too much unless it's a sick cell. So all the vesicles will just be split up into some set of destination compartments. Okay, so there's some sort of flux of coat gamma going from compartment J to compartment I. And what is that flux? That flux has to depend on whether the vesicle contains a factor which likes a corresponding factor on the target membrane. Okay? Is the stamp I have on my package the correct one that matches the address of the house I'm delivering it to? Okay? That also you need another matrix. So this is called the fusion matrix, and it just says that if there's some factor alpha on a vesicle and there's some factor beta on a target compartment, F alpha beta tells you how much the vesicle likes fusing to that compartment. And you sum over all possible values of alpha and beta. Okay? There's another exponent that arises here which has to do with the cooperativity of molecular binding. Right? And of course, the rate at which you fuse to a destination compartment will scale with the area of the destination compartment. And once again, I'm putting another exponent over here to keep things slightly general. Okay? This is a sort of conservation law which says that all the vesicles that come out from here have to end up fusing somewhere. So you just sum. Okay? Finally, this is the real conservation law. These are just fluxes. Okay? The flux of membrane from compartment J to compartment I, from compartment I to compartment J, tells you the rate of change of membrane area of compartment J. Similarly, the rate of change of this particular cargo on compartment J depends on how much of it is coming in minus how much of it is going out, and then you just run the system through. Okay? So I'm spending a lot of time going through this because it's nice to see exactly what the internals of some of these models are. Are there any questions?
Does a cell actually optimize the distance traveled by these vesicles?
No, because the cell has to move stuff from the inside to the outside. So distance is not the right measure. But time you could ask, I don't know. Yeah. No. The concentration of proteins on this particular compartment is X sub, on compartment J, it's this particular number. On a different compartment, it will be very different. That's the whole point, right? These are the state variables of this dynamical system. Right? Every compartment has a different composition. Okay, good question. Yeah.
So the alpha and gamma label protein.
And
therefore, which of these components, B alpha gammas are non-zero and X are non-zero, is basically which
proteins like to
interactions are contained.
exactly.
And those are the matrices that can in principle evolve.
Yes. And can be measured using cell biology or biochemistry. So unlike that last pedagogical model, this one I can actually measure things. Right? Okay, good. So here's what the model actually does. So you asked about these matrices, right? So these matrices tell you, do these coats like these particular cargo molecules? If the number is large, I've represented it by a big circle. If it's small, I've represented it by a small circle. In principle, there could be not a 1-0 interaction, but a whole quantitative interaction. Similarly, the binding of one vesicle to another destination compartment could depend on various affinities, which could vary between 0 and 1. Okay? So again, remember which molecules we're talking about. RABs and coats and cargo will segregate, and coats will segregate the cargo. And these proteins called snares will determine the fusion, just if you want to go read about it later. But here's the interesting thing that happens. Turns out that if you start the model and you have a lot of different compartments in the system, a lot of different compartments of arbitrary initial composition, it ends up just through the dynamics that they have eventually the same final composition. Okay, so this is an unusual feature. It's not obvious why this should happen, and we have some explanations for it. Okay, so in a real cell, this is actually exactly how it is. If you look at the Golgi apparatus, it will be spread in different parts of the cell in different compartments, but they all do the same thing. But if you look at lysosomes, there'll be many copies of lysosomes, but they're all doing the same thing. Right? So this model actually recapitulates the behavior of these cells. In fact, by changing these exponents, it's actually possible to make all kinds of things that look a bit like real cells. Right? You can make two different organelles, which are just one compartment each. or you could make two different organelles which are made up of a set of compartments that are all the same size, right? Or you could have something arbitrary in the middle. So this is sort of a degenerate state. Okay? So this model actually does something moderately interesting. Now, let's see how the dynamics goes over time. In this case, I have a system which contains only two types of coats and two types of snares. Okay? And therefore, the composition of every compartment depends only on how much of snare 1 and how much of snare 2 it has. Yeah? Mu and nu, this was the exponent for the budding, how proportionate you are to the budding area and how proportionate you are to the fusion area, and this is the exponent of that. Yeah? And this n is the cooperativity of specificity when vesicles fuse to targets. Okay, so here, watch what's going on. I have a cell, and in this cell I have 10 different compartments. That's why there's 10 different initial conditions, which are the ends of these little threads over here. Okay, so I have a cell, I have 10 different compartments of arbitrary composition. As you run the dynamical system, they exchange matter in these fluxes, right? So their compositions will change over time, and this is how they change. But eventually, all the compartments in the cell end up having the same composition, in this case, which is a sort of a boring cell, right? However, if my fusion, this cooperativity number, which maybe we can discuss later, it's basically how specific you are in your molecular interactions, how much I like the people I like versus how much I don't like the people I don't like. If that is high enough, then what happens is something different. I again start with 10 compartments of arbitrary compositions, but they end up with two different compositional subsets. Okay? These are not different fixed points of one dynamical system. Okay? This entire thing is the fixed point, which happens to be characterized by having two different compositions. So finally, I've got to the end of what I wanted to say. Remember, I set up this problem. I said, how can changes in the genotype of an organism, particularly this gene family expansion that happened 2 billion years ago, lead to having more compartments? Here's how it can happen. So I'm going to walk you through this. This is sort of done by hand. I'm going through a particular scenario the way it might have been. I'm not saying this is how it happened. In my scenario, here are my fusion and budding matrices. The circles are large if the entries are large, the circles are small if the entries are small. Okay? Here I have a system with exactly two types of snares and two types of coats. But in this case, this particular snare likes this one, but it also likes the other one. So all vesicles will fuse to all the other compartments, right? So eventually, all 20 compartments in the cell end up with the same composition. However, if I now make, through the process of evolution, this snare dislike that one, or this snare dislike that one, in other words, suppress the off-diagonal terms, then you end up with all these compartments becoming this type and all these compartments becoming this type. So here you have a cell with 20 compartments, but it ends up with two different organelles, which could be the ER and the Golgi, for example. Similarly, if I have 3 snares, right, then every compartment is described by 3 variables. Of course, I'm not going to plot a 3-dimensional thing here. So I've just compressed the trajectories in 3-dimensional space using principal components. But in any case, these are compositional trajectories of every compartment in the cell. And you start off with 10 different compartments. They end up with 3 different types of states, right? 2, 3, and so on. Now, this is where the evolutionary scenario comes in. Imagine that you duplicated this particular snare. So now you have 2 copies of it in the genome. Both the copies are identical, so they all like to talk to each other. If they all like to talk to each other, there's actually only really 3 different molecular varieties there. And as you would expect, they end up making 3 different types of compartments. Now, over time, evolutionarily, if these off-diagonal elements become smaller and smaller and smaller, in other words, if the duplicated genes end up taking on new roles, this is called duplication and divergence. It's a very common theme in evolutionary biology. Then, indeed, duplication followed by divergence takes you from a cell that has 3 types of compartments to a cell that has 4 types of compartments, which is basically the problem I set up in the beginning. Okay? So this can actually happen. I'm going to skip over this slide because I don't have time, but you can actually show that a cell that needs more and more compartments actually have to make the off-diagonal terms smaller and smaller and smaller. But it's not such a huge thing. I mean, 10% error rate is actually something one can accommodate. Okay? So, by this time, you guys should all be saying something's wrong with this. Okay? Because in my evolutionary scenario, I've said the following thing. If I want to take a cell with 5 compartments and make a cell with 6 compartments, right, I have to duplicate some set of proteins. What proteins? All the proteins that you need to make a compartment. I have to duplicate the snares, the RABs, the coats, the tethers, all of them. But once I duplicate them, I have to make sure they don't talk to each other anymore. In other words, they have to stop talking to each other for the cross terms, but they have to continue to talk to each other inside. So this is evolutionarily a completely unlikely scenario. Right? So that's what I've shown over here. You have a cell which has two compartments and two types of coats and snares. Right? If you duplicate the genes and you put them back in the same cell, you still end up with two compartments because all the proteins still talk to each other. Right? What you need, ignore the third column here, what you really need is somehow for these off-diagonal terms to be suppressed. There is an easy way to do that. Okay? There's an easy way to duplicate a lot of genes at once and to stop sets of genes from talking to each other. And the way that might happen is, you might think this is cheating, but I'm going to do it the following way. I'm not going to duplicate the genes inside a single cell. Instead, I'm going to allow this cell to become two different cells. Right? So now you have twice as many genes, but they're in two different cells. Now I'm going to allow the cells to evolve over 20 million years, 100 million years, whatever it is. Right? So of course, these guys will still be talking to each other. These guys will still be talking to each other, but these will stop talking to this over time, just because you need a certain evolutionary pressure to maintain interactions. Suppose now I took these two cells and stuck them together. Then I get exactly the kind of interaction matrix I need to make new compartments. Right? So I'm making the claim that if you take two cells that are very distant and you hybridize them, then you end up having a cell that has more compartments. And this nicely solves all the different lacunae of this particular model in one stroke. Yeah. How is it different from endosymbiosis? It's not, but endosymbiosis happens rarely. But you could ask how often hybridization happens. And so it turns out hybridization happens quite frequently, and one of the best studied examples deals with this organism that was isolated. I was in Copenhagen last year. This is a microbiology lab at the Carlsberg breweries. This was the lab of a microbiologist named Emil Christian, and he was the guy who isolated the yeast that makes lager. So if any of you drink Kingfisher, Kingfisher is a lager type of beer. It's the sort of paler type of beer. That species is a species of yeast, which used to be called Saccharomyces carlsbergensis, but it's now actually called Saccharomyces pastorianus. Okay? So it turns out that that particular organism is a bit of a freak. Okay? So Saccharomyces pastorianus is not the same as the yeast that makes wine, which is Saccharomyces cerevisiae, which is also the one that makes bread. Okay? If you look at the genome of pastorianus, it's very large. It's much larger than the genome of cerevisiae, but it contains half its genes are very, very close to the cerevisiae genes. Right? So in other words, this species, for a long time, it was known that it was probably a hybrid of cerevisiae and something else. Right? So over the last several years, Diego Libkind and colleagues have been roaming around the earth looking for the other parent of this particular hybrid yeast. And in 2011, they finally published that they had found the other parent. They found it in Patagonia, South America, down there. That particular yeast is cold tolerant. Right? It grows even in the winter in the ice. Right? It survives. So it turns out that to make lager, you have to brew at 10 degrees Celsius. So this species has to be cold tolerant. That one's not cold tolerant. That one is, and somehow these two got together. How could they possibly have gotten together? Because cerevisiae is a European wild stock. Eukaryotes is a South American wild stock. Well, 500 years ago, Europeans started to go back and forth between here. They were making ships and barrels out of oak-like trees over there on which this creature lived. Right? So we can actually date the time of this hybridization to the time of trafficking between these two continents. Right? Incidentally, the origin of lager is about 50 years after the first documented movement of ships between these two things. So it's very, very tight. The more interesting number I like is this one. These two species turn out to be separated by 20 million years, hugely diverged. Right? So this is a hybrid between species that are about as unrelated as humans and lemurs. Right? Two very different things. Yeah. Not over 500 years. They hybridized in an instant 500 years ago. It happened once. Yeah, good question. So this organism, so cerevisiae has about 6,000 genes. Eukaryotes is also a Saccharomyces. It has about 6,000 genes. Their genes are diverged by about 85%, which is how I'm making this claim about the difference between humans and lemurs. But the Pastorianus content that comes from cerevisiae is 99.5% identical. The content that comes from Eukaryotes is 99.5% identical. In other words, this thing is truly just a mix of two different creatures, which is basically what I needed for my model to work. Right? There are 3,500 genes present here that are present in two copies, although some have been lost. Okay? And finally, if you look at the trafficking system, if you look at the snares, the RABs, the coat proteins, okay? If it's shown in white, that means it's present from both parents. If it's shown in blue, it comes from Eubionus. In pink, it comes from Cerevisiae. So the point is that the trafficking system of this hybrid organism is actually the type that was there in the model. It's something which contains components of two very different protein sets, right? So it's, entirely plausible that it contains additional compartments. But of course, to check this, one would have to do experiments, which we are planning to do. So I'm going to close with this. So I've shown somehow, at least I've tried to indicate, that the origin of eukaryotes is a sort of murky subject, right? There are many different scenarios that people have put forward to explain archaea, eukaryotes, bacteria, and what we see today. We can't use cellular evidence to distinguish between any of these hypotheses because those intermediate cells no longer exist. They're all dead or we haven't found them. We can use molecular data to separate these different hypotheses, and that's part of what we are trying to do. But there's one more thing we can do. We can make an assumption. We can say that there could be evolutionary processes going on today that are similar to what happened during eukaryogenesis, right? And therefore, by doing experiments on evolutionary cell biology today, you could at least constrain some of these models. You could eliminate some and say that others are more plausible. Okay? So in other words, the evolution of eukaryotes has finally turned out to become a problem that is experimentally tractable today in the lab. Okay, so my time's run out, so I'm going to close. You can read these. Soumya, who's sitting in the audience, is better than me to tell you about these binary traffic models. Ramya is a student in my lab who's doing the lager yeast evolution experiments, and you can read these papers. So, and again, of course, you can contact me if you need anything. So, if there are questions, I'll take them now, and then we'll have to stop. I mean a new organelle, which has a completely different composition than any existing organelle, to which I can target proteins completely independently of any existing organelle in the cell. All organelles bud and fuse vesicles. All, but they don't share with all the others. But eventually, they has to cycle. So there are directed cycles that will carry you from any organelle to any other organelle. At worst, they'll go through the. Sorry, I'll take that one in the back.
Can there be kind of phylogeny of these organelles?
Very good question. So is there a phylogeny of organelles? That's part of the things you would like to address. Maybe you could show that somehow the lysosomes and the Golgi are both descended from the ER. And this kind of argument would seem to indicate that is the case. The problem is that these things are so ancient, they predate LICA. So you want to do phylogeny of proteins that go over 1 billion years. And current techniques do not allow you to do that. That's the problem. But it's a very good question, yeah.
So just you're trying now experimentally to look for if there were different compartments in this.
Yes. Yes. Yes.
And would you define a different compartment as, let's say you had two, probably one had
This type.
different proteins on the outside, the other one, then you would say, this is new compartments, or would you say it has to be something not?
No. So the new ones that we have will be the same flavor as the ones that we had before, right? But they will be different. But what I would do then is, suppose you find something like that. What you would do is take the subset of genes involved in that, put it back into just Cerevisiae, and see whether you can create one new thing, not all things in pairs, right? To do that without knowledge of evolution would be impossible. I'm just trying to use evolution as a guide. Was there? Yeah.
So initially there was this prokaryote,
Yes.
and it was taking food, and they would double.
Yes.
Now this, say, parasite
Yes.
gets into it.
Yes.
Now for the whole system to double, you need more food, right? But the food that is entering depends upon the surface area of the first.
Very good. No, so these things are not, so eukaryotes are actually much more efficient at taking stuff across the membrane than prokaryotes are. So membrane is not limiting.
Membrane is not limiting, but I mean there better be some change in metabolism itself, metabolism as well, because now you need more amount of food for both of them to double.
That turns out not to be the case. Yeah. You might have thought about that. Yeah.
Question might not really make sense, but so we see multicellularity in eukaryotes, but we don't really
I'm glad somebody asked that question. Okay, so why does multicellularity fit into all this? The answer is it does not. So let me go way back. I hate skipping over slides like this, but turns out it does not. Okay, So these are just eukaryotes. Let's talk about multicellularity. Multicellularity has arisen, true multicellularity has arisen in plants and in animals. Okay, so that's just 2 data points on the eukaryote side. Okay, close to multicellularity has arisen, of course, among the fungi and various other types of eukaryotes. Right, close to multicellularity has also arisen among the bacteria. You could talk about bacterial consortia or communities. Right, so if you talk about 2 data points versus 0, that's not enough statistics. But if you talk about close to multicellularity, it easily happens on both sides. You can actually evolve multicellularity in yeast in the lab today. So it turns out that multicellularity is an easy and obvious step in evolution. Which happens repeatedly all the time and doesn't require any special explanation.
Also slightly tangent to what you said. So you were trying to make a case for how organelles come.
Yeah.
Are generated. But we now know that different organelles have different functions.
Yes.
In sort of very cell biological sense.
Yeah.
So is there sort of a class of 10 functions which get segmented? How many organelles are there?
Yeah, so even to count how many organelles there are is sort of ambiguous because they're not in steady state and so on. And you could ask why you need organelles and whether having organelles makes you successful. So, I've left function out of this altogether. I would say that we have to answer that question. And I would say pick a particular model where it's tractable. And if you are interested in such a thing, I would suggest going and looking at the Golgi apparatus and asking why it needs n different cisternae. That's a very focused way to ask the same question. But yeah.
Yeah. So in your biophysical model, you've assumed that the rate at which vesicles are budding off is proportional to or some function of the area of the compartment.
Increasing function of the area.
Yeah. Area of the compartment.
Yeah.
But it could, in principle, also depend on what the cargo is, right?
It does in this case. So the cargo can actually recruit different coats at different rates. So the dependence on cargo is part of this.
No, and I mean, what I mean to say is basically it would, for instance, if it's some protein that is getting transported, then the rate at which a vesicle buds off depends more on how often that protein is required than what the size of the compartment is. Right?
Required? You mean in terms of actively controlling the flux of the vesicle?
No, in terms of whatever function it is supposed to perform.
Yeah. That's called signaling broadly, and we've sort of left signaling out of this. This is the constitutive behavior of the cell. Such things are well known. There are many examples of what you just described.
Okay.
So, because it contains this cargo, that cargo it binds to the correct motor protein, that motor protein takes you to the right destination. Yeah, sure. It's all molecular interactions, all the way down. Sort of well understood. Last question.
So, in a given kind of cell that exists today, you have a list of the snares,
Yeah.
the wraps,
Yeah.
all that
Yeah.
stuff.
Yeah.
You know which ones interact with which ones.
In many cases.
So, effectively, you can construct these FNG matrices.
You can.
FNG matrices.
You can. Not completely. You know some of these, not enough of them.
So, based on a construction, whatever
Yes. Does this work?
of these FNG matrices, the model then sort of predicts the compositions of compartments that
Yes.
you're going to have, right?
Yes.
And can that be tested?
You would have to make a model that has many more layers than this if you want to do that level of quantitative prediction. I don't think it's currently plausible.
If not composition, at least
Numbers.
presence or absence of,
Yeah. So the presence or absence we can start talking about. Right? But composition varies. It's a bit dicey. Last question on the, in the
Okay.
back. Yeah.
I was just wondering, are there any studies that are being done on conservation of these organelles across different eukaryotes?
Yeah. Sure.
So suppose, say, I take a mitochondria out of yeast and put it in a mammalian cell, can it support it?
No, so that won't work. But certainly there are organelles that are present in diverse eukaryotes. The problem is you can't just transplant an organelle because the organelle doesn't bring its own DNA. Okay, mitochondria do, but they also need the host genome to interact with it. Okay, so just moving an organelle into a cell doesn't help. One important and interesting thing is that many lineages of eukaryotes have organelles only that they uniquely have, that others don't have. So there are new organelles. That's also there. Another interesting important thing is there are eukaryotes which are cannibalistic, or they'll eat other eukaryotes. They'll destroy the membrane of that eukaryote, and they'll keep the organelle of that eukaryote for as long as before they divide. So for example, they'll eat chloroplast containing organisms, and then while that thing is still in there, it can photosynthesize. But once it divides, the plastid goes into one cell, the other one has to go eat another alga. So, organelles can live in the context of other cells. Okay, so I'm going to stop here for lunch, and also because there's an ICTP ICTS MOU that's being signed next door. So and we can discuss this later. So after lunch, we meet again at 2 o'clock here. Boris Schreiman will give his lecture, and remember at 4 o'clock there's a tutorial. Okay, good. I don't think you should clap. So let me take off this mic.